Hello. I would like to do a quick demo of how to use PDB for the assignment in this class. And so we're going to start with just PDB website and search for ligand. So I'm going to do propafenone, the drug that I'm interested in. This quick search comes up with only one structure. And each structure in PDB has a unique ID or accession number. So this one is 7FBS. If you know this unique ID, you can always find the structure again. And then there are two tabs we're going to pay attention to. The structure summary is kind of an overview of the structure. And then the structure itself is where we're going to look at intermolecular interactions. Okay, so just to kind of get a quick overview of this entry. So you have a quick view for the structure itself. Notice this is biological assembly, which is kind of what the best guess of the authors is, what this protein looks like when it's doing its function. The asymmetric unit for this structure is the same as biological assembly, but it's actually more of an kind of procedural for solving the structure. You can have proteins crystallize in an asymmetric unit that have like multimers, multiple proteins pack into the asymmetric unit. And so do not trust anything about like asymmetric unit. If they were like, if this was a tetramer or octamer or dimer, doesn't really mean anything, right? So we are more interested in biological assembly. And so this just says it's a monomer. That's why there's not really any discrepancies. And so to begin with, we have some parameters for the structure. You can ignore that. The important part here is the reference, who created this model, right? So these are three-dimensional models of proteins, and has it been published? So there's a article here from Cell. So these are the authors with the original publication. And if you have questions about the structure, you can always click on it and go to PubMed or the original source to actually like read the article about the structure. Then if we scroll down below, we have a list of the actual macromolecules. So, we're focusing on proteins, but there could be lipids, there could be carbohydrates, right? So they're separate. And each component of the structure model will have kind of a special label, right? So here we have chain A, it's a monomer. We have a little bit of information about this polypeptide chain, right? And the chain A, that's what the authors call it. So sometimes there'll be some discrepancies in how, in general, protein like chains are referred to and how the author calls it. So always good to pay attention. And so UniPro is the big database for the protein. So we have links to that, like what this protein looks like in that database, and you can link there directly, again, with unique IDs. And then I guess the sequence overview is basically taking this polypeptide, stretching it out, right? So this one is a really large one, more than 1,800 amino acids. And then information about different properties are laid out as tracks. So you can see in the UniPro, right? So we have two IDs, and it seems like in UniPro, there's two components of these proteins in separate entries, right? And that kind of makes sense that the first one, this P15389, that's the red protein, and the other is is a GFP tag, basically. So this protein has been tagged with GFP in the last 200 amino acids, right? So the one that we clearly care about is the one up here at the front, okay? Then unmodeled means there are some amino acid residues that don't show up in our three-dimensional model. And then this part might be important, the mutation. So this protein contains a mutation. And if you have a mouse with a scrolly wheel, you can just kind of zoom in on it and see where that mutation is, right? So in our protein sequence, there's three glutamines, QQQ. That's actually a mutation. And in the original protein that's in the UniPro, it's CFT, right? So we know this is a mutant protein. And then, like, if you're interested in PFAM, if if this protein has entry in protein families, right? There's a link to that, shows where the PFAM uh, entries are, domains. All right, so then besides this was our macromolecule, so this is just one protein chain. If your protein has multiple protein chains, you have all unique chains laid out this way, and it will say if this was a homodimer, two of identical chains, you'd have chain A and B, but they would be listed as identical, right? But if it was uh, heterodimer, so chain A would be listed separately and chain B would be listed separately as well. Okay, then when we get down to ligands, there's four different kinds of small molecules that are that are present in the structure, and you can read about them. And some of them uh, are showing up in multiple copies, right? So notice they have their own IDs for like this structure. And these are listed as chains, even though they're not polypeptide chains, right? But like for the purpose of this model, I, H, G, F, and E are copies of this particular lipid molecule, right? And makes sense if we're looking at like a channel structure that the lipids are going to help it kind of keep the shape. Okay, so propafenone, the drug we're looking at, I can just recognize it because I looked up its structure. It's right here. It has kind of a longish name. And what's nice about this link, I can just go directly to the interaction. So if I just click on the link, it will take me into the structure, focusing on this ligand and showing what are the nearby amino acid residues. Okay, so this is a direct link. Uh, Alternatively, I can go to the structure tab, and the structure tab uh, gives me amino acid sequence, and I can change if this was multiple polypeptides here. I could change which structure is displayed here and which one to focus on. And then we have the actual structure display that you can rotate with the movement of the mouse. You can also press I think on the Mac, let's see, I think Control and click with the left mouse, you can move the structure around to recenter it. And then the other interesting feature that's useful, when you point to some part of the structure, at the bottom, we get the tag that tells us what we're pointing at. Like, for example, this is chain A, alanine 457, and notice in parentheses, author 744. So that's actually really useful to note. So the author calls this residue is like number 744, while in a typical structure, this it would have a different number, 547. So that's because when proteins are expressed, they often have tags, N-terminal tag or C-terminal tag, which is additional amino acids residues added. And so try to keep track of kind of what you're looking at. Ideally, do keep track of both numbers. Okay, so I can look around. It's a little tricky here, right? So like all these lipid molecules are folded around this, and it's a little hard to find mine. I can see it right there just because I've looked at this before. But again, your shortcut could be much more easily to go right from the summary page. So I think I see my structure right there, and you can see on the bottom when it displays it, this is like chain O, and let's see, where is it? There we go. But author calls it A. Weird discrepancy. Okay, so if I just click on it, you can see small halos show up around the structure, and that just means this is selected. It's also reflected up here in the control panel that part of the structure that's right now in the focus is this ligand O. And what we're going to do is, this looks really cluttered. Right? There's like a lot of stuff is showing up here. So I want to do a couple of things. So the reason I'm focusing on this, I'd like to know how this ligand interacts with its protein. So I'd like to be able to see different non-covalent interactions. And so what I need to do is control what kind of interactions are displayed. Just by default, the displayed ones are hydrogen bonds. And again, if you point your cursor to it, on the bottom, you can see like here's hydrogen bond between this and that, right? So, and this actually is just between amino acid side chains of the polypeptide, right? And I'm interested in my drug interacting with a protein. So this would be an example. So glutamine 372 interacts with my protein, with my drug. Here's the side chain of glutamine. But this looks really messy and hard to see anything. So there are two control boxes that we're going to use. And let me also just recenter my structure a little bit. So remember, click left mouse with the control allows me to translocate the whole structure up or down. All right, so I want to focus on this, but I want to remove extra clutter. So within the display window itself, there's this little slider kind of a window that allows me to change clipping. And so you can think of clipping as how thick a slice that you're looking at. And so if I increase it, it's going to remove things outside, like of where I'm focused on. So maybe this is how far I want to like clip it. Well, so the scrolly wheel on your mouse also allows you to change it, right? So now if I look around, it's like we're only focusing on a thin slice, right? Think of like deli slicing your turkey or whatever deli meat you eat. You can have thick slices or thin slices. And so this I might need to like change my slice a little bit so I can see the whole structure, but it's pretty good. Like I can see the whole structure here without too much clutter. Okay, so that's the first one, is changing the slice, how thick of a slice in space, and everything else is peeled away, removed. Okay, the other one we want to look at is right here. So if you click on this set of controls under components, then down at the bottom, you have non-covalent interactions. And you can see the defaults are set to show hydrogen bonds and a few other interactions. So I'm going to just deal with them one at a time. So I'm going to start with ionic. Are there any ionic interactions? No. Right, so nothing is showing up, so I have no ionic. Pi stacking, also no. Cation pi, don't see anything. Halogen bonds, I'm going to skip. And so when I add hydrogen bonds, that's where I can see, here's the hydrogen bond, right? So my structure has a carbonyl here that has hydrogen bond to glutamine 372. And so I can just rotate it to show kind of this interaction. I can zoom in a little bit and maybe like take a picture. Let's see which way. Oh, so maybe like this view. It's a little bit, pushed back, but it seems like it's a good way to show it. Oh, so maybe I need to make my slice a little bit less, like a little bit less thin, so I can see more stuff, right? So go back to this side. Oh, perhaps like this, right? So here's my glutamine. I can clearly see the side chain with the amide on it, and I can see this. So at this point, I can just take a picture of it, right? And maybe like this. You can take a screenshot just using this little shutter screenshot and download that screenshot. Okay. So then let's kind of move on and see what other interaction. Seems like only one hydrogen bond, right? But the reason we're thinking about this is think if a person carries a single nucleotide polymorphism that changes as glutamine to something else, right? Say to alanine, can you see that you would then have much weaker binding of this drug for that person's protein, right? Because if only alanine has CH2 group, then you would lose this hydrogen bond. All right, so I'm going to move on. Weak hydrogen bonds, I would say let's ignore them. Not really sure why they're showing up. We can talk separately about that. And then when we add hydrophobic, that allows us to see what kind of hydrophobic interactions this protein, this ligand has. And you can see there's quite a few, right? So then we can just basically explore one by one what they are. So this group right here has two links to two hydrophobic interactions, and I can see what it's linking to. So that's another ligand, right? We're not going to do that. That's another ligand. Okay, we're not interested in those. And then on this side, I can rotate and see that my is probably benzene ring, right? Links to phenylalanine 403. Right, there is another phenylalanine 1444, and just kind of look around somewhere else. And so then take a few pictures so you can say, well, what is the protein requirement for binding this ligand? Thank you.